And thank you very much, UPLB MNH, for having me here. I think this is a very good opportunity for me to share what we're doing in relation to marine mammals of the Philippines. And I'm very happy to have you here right now because later on I will be calling for some volunteers to help us in some of our efforts. And also I like that we are an intimate group here, so you can be comfortable in asking questions later. Okay, so sabi nga ni Dr. Diliman saga na to, actually she's already familiar with some of my slides because what I do is that I always introduce what the general program is all about, and then I just give updates and highlights of our findings. So let me start by proudly saying that our country represents 1/3 of the world's cetacean diversity. So we have 28 cetacean species. That's out of 30 marine mammal species in the Philippines. I think most of you are already familiar with this group. So as a group, of course, we all know that marine mammals is a collective term used to refer to animals with adaptations to aquatic living. So it's not a formal taxonomical grouping. This is actually made up of different species coming from different ancestors. So this is a polyphyletic group. So we have 3 orders under this. So that means these orders have their aquatic lifestyle. Okay? So we have order Cetacea. So the order Cetacea is actually comprised of 3 suborders. So we have the Archaeoceti, the extinct species of whales and dolphins. Then we have the suborder Odontoceti. So that suborder is comprised of the toothed whales and dolphins. And then we have the baleen whales under suborder Mysticeti. And then we have order Sirenia, which in the case of the Philippines is represented by our dugong. So it has 2 families, the Dugongidae and the Trichetidae. Okay. And then we have order Carnivora, comprised of piscipedes and pinnipeds. Okay, so these are the 28 species in the Philippines. So these are the odontocetes, meaning they are provided with teeth instead of baleen plates. And then as to Sirenians, we represent the dugong. Okay, we don't have manatees here in the Philippines. And I think you're already familiar with this. So there is controversy with regard to its taxonomic classification. Okay, so I think you have read articles about discussing about its genus. Okay, so we have this flawless otter in Palawan. Okay, so please take note that among the 30 marine mammal species in the Philippines, 6 are considered either threatened or vulnerable according to national and international listings. So we have the giant sperm whale, queen whale, humpback whale, Irrawaddy dolphin, dugong, and blue whale. So we are using cetaceans in the Philippines as sentinels of One Health. By sentinels, we mean animals that can indicate the conditions of their habitats in measurable parameters. While we can use other vertebrates as sentinels, for instance, we can use fish species. There are many traits of marine mammals that we deem important for them to be used as sentinels, not only of ocean health, but also of human health. For instance, we share a lot of physiological mechanisms with them. There are many studies right now presenting similarities with immune mechanisms. They are also known to have relatively long lifespans. They migrate, okay? And they are able to accumulate toxins, biological pollutants from the environment. So I think you've also heard about One Health. So this concept is actually an emerging concept in the field of research, which is now calling for interdisciplinary efforts. in trying to elucidate the connections among ecosystem health, animal health, and human health. So the role of cetaceans right now, worldwide, is that a lot of species are being used to indicate chemical pollutants, biological pollutants, and the impacts of human and fisheries interactions. So we have this ongoing effort. We call it Philippine Marine Mammals in One Health, in which we are using cetaceans as sentinels of marine zoonosis. Of course, you're already familiar what zoonosis are. And we have also our researchers using cetaceans as sentinels of fisheries interactions. This is actually a collaborative effort among mostly government sectors, government agencies, and we are collaborating with the Natural Sciences Research Institute of UP Diliman. We have 2 laboratories there, the MRSL, Microbial Research and Services Lab, and the Molecular Protozoology Laboratory. My lab, METAS, as mentioned by Dr. De Leon, it stands for Microbial Ecology of Aquatic and Terrestrial Systems. And then we have the Marine Mammal Research and Stranding Laboratory. We have also a very important partner, the Bureau of Agriculture, specifically, the Department of Agriculture, specifically their Bureau of Fisheries and Aquatic Resources, the BFAR. Then we have an NGO partner, the Philippine Marine Mammal Stranding Network. And I also talk to other persons involved in other NGOs, because we want to collaborate with other sectors. Okay, so our samples are coming from stranded individuals. You know where to find marine mammals. Please take note that most of our cetacean species are pelagic, so they stay offshore. So if we want to sample their skin, for instance, if we want to sample their blubber, it's very difficult for us to do that. So we need a lot of research resources. We need a dedicated research vessels. We need researchers who can stay offshore. And it's very traumatic for the animal because we need to capture them just to get the samples. But for skin and blubber samples, for instance, we can use dart guns with buoys. Then you can just retrieve them later. But it's very costly. And it has been observed in the Philippines that there is an increasing trend of stranding events through the years. And so we are maximizing those events by using stranded individuals as samples. So what is a stranded cetacean? So I have here the technical definition: any dead cetacean on shore or any live cetacean cast ashore or unable to return to its natural habitat. Every stranding event deserves scientific attention because it is not normal for any cetacean to just strand without any important reason. So why do marine mammals strand? There are many purported reasons. So right now we are observing a lot of cases where stranded individuals get entangled in nets, okay, victims of fisheries interaction. Extreme weather events, earthquakes, typhoons, beach topography. It has been observed that there are many cases of strandings in sites wherein we have shallow beach topography, just like some sites in Australia and New Zealand, for example. Predator interaction, you can see there the damaged body part. And we have also shark attack, which is actually a predator interaction. Diseases, okay, infectious diseases. For instance, we have cases of Brucella infection, Toxoplasmosis infection, viral infection in other areas, especially in North America. Human debris, okay, that is a sperm whale. You can see at the left photo, okay, very big plastic, 2 meter by 2 meter, large in its respiratory tract. So, ang laki-laki niya. Nakakaawa. Okay, pag ni-rescue mo and then hindi na siya makasurvive, hindi na makakain. And then, when we open it, may plastic sa loob. So in general, it's really difficult to determine the specific cause of stranding. When we encounter a stranded individual, we cannot readily say if it's due to illness, due to plastic. A lot of cases, we observe a synergy of different causes. But it's really difficult. We are using a lot of evaluation protocols, but until now, I think we have only confirmed less than 20% of cases as to the cause. Okay, so I would like to share this data. So from 2005 to 2018, that is until August 2018, we are still updating the database. We have a total of 885 cetacean stranding events in the Philippines. So that gives 65 events, average of 65 events per year. You can see here the increasing trend, which is actually also, or maybe we hypothesize that the increasing trend may also be the result of increased effort of reporting and observation. And based on the distribution of the stranded species and individuals, the data can also be an artifact of the distribution of cetacean species in the Philippines. Until now, we don't have comprehensive data regarding the distribution of our cetacean species. And we also have the so-called hotspots. So these are the administrative regions in the Philippines where you can, where we have observed a lot of cases. So in general, we have the top administrative regions, Region 1, Region 3, Region 4, Region 5. So most of the events are reported from those regions. So what do we do during stranding events? So we usually respond to the animal first, okay? Sampling for biological materials is not a priority when there is a stranding event. We need to consider the disposition of the animal. We need to consider first if the animal is stressed. We need to consider first what kind of help do we need to give to the animal. So usually nagsasample kami second day na, depende sa kondisyon ng stranded animal because the primary goal is to help the animal. Okay? May mga namamatay rin na stranded individual during these events. Stress, myopathy, for instance. So that's why we also follow standard protocol. That we don't want a lot of people there. We don't want media first. We want first to stabilize the animal. And then when we are sure of its condition, that's the time that we can try getting blood samples, for instance, getting swab samples, for instance, getting condensate from blowhole exudate, for instance. So if you want to deal with cetaceans or use stranded cetaceans as samples, it's actually difficult. It's a way of life. It's a calling. You need to help them first before you can get their materials for your research purposes. And you can't do it alone. You need to collaborate with other stakeholders. So in general, we gather two types of data: biological characteristics, the size of the animal, the skin color, okay, the shape of the fins, for instance, because we don't know yet, okay, a lot of species. There are first records of species coming from stranding events. So hindi natin alam that these species are already here in the Philippines, pero nalaman lang natin when we observe them during stranding events. So that is also one of the reasons why we consider the stranding response as very critical for the conservation of our marine mammal species. As the stranding characteristics, we also gather information from the locals, for instance, the time, the specific coordinates of the place, what information they can offer about the event, okay? And then we respond to live individuals or freshly dead individuals. So we have there the technical definition of a freshly dead. Kasi kapag decomposing na, of course, you don't need to help the animal anymore, and you can get a limited number of samples depending on your purpose. Okay? So to standardize the response, we have this so-called Stradin response report form, which has been developed, critiqued, revised, improved, and through the years, okay, through collaboration with many stakeholders. So until now, we are still revising our protocols, and we are trying to contextualize it, considering the logistics here in the Philippines. So a lot of islands, archipelago kasi tayo, di ba? So ang hirap. So what if the stranding event is in Ilocos, or in Davao, or in Mindanao? That's why our researchers are always ready. So we usually advise them to have a backpack ready. So if there is a stranding event, you need to go to the site. So you need to be on call 24/7. Okay? So whenever students apply, okay, for mentorship, or if they want to participate in the research efforts, sinasabi ko na sa umpisa pa lang, are you ready for this lifestyle? Okay? Pero wag kayong matakot, baka naman kasi may interested. Fortunately, we have many collaborators. Ikikwento ko sa inyo later, okay? What's happening in the field. Okay? So, we have this one effort. So, we are observing cetaceans as sentinels of human interaction. So, under human interaction, we have categorized different types of interactions. For instance, we have direct interaction, direct attack, okay, physical injury incurred by humans, for instance. Chinap-chop yung dolphin, for instance. Binenta sa palengke, okay? You can see this one. I'm sorry. Ulo na lang, okay? Putol na, okay? And we have also fisheries interaction under human interaction. So for this effort, I developed this protocol for observing cetaceans for physical signs of human interactions. So this is actually modified from the works of Gerasi and Lounsbury, if you're familiar with those authors, Reed and Murray, but we tried our best to contextualize it, considering the nature of our cetaceans here in the Philippines. So this is a sample form, okay, which are being sent to us by our collaborating veterinarians. I think you know that the Bureau of Fisheries and Aquatic Resources have provincial veterinarians, and they also have their fisheries officers. Okay, so they are the ones who actually experience these conditions in the field. And this is a form which was accomplished by another team, so you can see their data entries. Okay, so for instance, we are asking if there are parts observed for hemorrhaging, body condition emaciated or not, depending on the amount of blubber. So you need to really observe. Okay, so this is another form, okay, for internal examination. For instance, gross pathology, stomach contents. If you see wounds or abrasions, you need to identify in which particular part, what is the type. Okay, so we give trainings also, and they have their stranding response package. So we send the kits and packages to the provincial offices of BFAR to be used by the veterinarians and other volunteers. So this is a sample attachment which will guide the observer how to classify the individual, for instance, if it is live, dead, freshly dead, decomposing, etc. Okay? So, some sample information attached in the protocol. So, here are some of the cases which were confirmed to be caused by fisheries interactions. For instance, we have this rough-tooth dolphin with fishing hook. Nakita niyo pa yung fishing hook. Okay? Doon yun. Kumabit doon sa chan niya, sa stomach. Hirap-hirap. You can see a suffering animal. So we have tried our best to remove the hook. Sumuko rin siya. Okay? Then we have this one. So this is from Zambales. We have this one from Camarines Norte. Hook in crab pot. Okay? And this one from Alban and tangled in drift gillnet. We also try our best to identify the types of gears, okay, used in fishing activity for us to, possibly correlate our stranding cases with the nature of fisheries in the area so that we can also inform the local government units. So I'm actually focusing on microbiological researches, and I will be sharing to you our ongoing research efforts. So for the purpose of these researches, we have actually established a nationwide system with BIFAR and an NGO of collection, transport, storage, and processing of tissues. And I can say that we have improved through the years. So, nung ako yung nangunguha ng sample, talagang ang gulo ng buhay ko. Like ngayong week, kunyari, dalawa yung na-strand. Yung isa nasa Ilocos, yung isa nasa Bicol. So, pupuntahan ko yun lahat. Okay? Kaya nga, i-share ko rin to, napansin ko yung lahat ng photos ko during stranding events, para-pareho yung damit ko. Kasi pala yung backpack ko, okay, I have this maong, I have this cargo pants, I have this black shirt. Yun lang lagi yung nilalagay ko doon after. Doon napansin ko, oh, pare-pareho pala. Buti na lang may date yung camera. Okay. So you will go there, okay? Madalas hindi ko alam kung anong address. Okay? And when you arrive at the site, syempre riot yun. A lot of people wanting to see the animal, may media pa, you need to talk to the mayor. Okay? So you need to connect to the community as a scientist. Di mo pwedeng sabihin, alis kayo dyan lahat, magsasample ako. You need to explain to them. So in every stranding event, we need to do that. We need to educate the locals. We need to debrief them, for instance. For instance, when you open the dolphin, you can see a lot of plastic there. You need to debrief the young, for instance. Kasi yung iba, shocking talaga yung mga nakikita nila sa loob. Okay, so that's only one way that's also one way for you to share your expertise to the community. But right now, we have this system of collecting and transporting tissues participated by the veterinarians and fisheries officers. So our partner NGO and BIFAR have sponsored a lot of trainings, actually hundreds of trainings already. And my participation in that training is that I am the one providing the customized microbiological protocols. So I prepared the protocol also in collaboration with other scientists and researchers as stakeholders. So this is a sample kit being distributed nationwide. So if you're a veterinarian, for instance, or a biologist, for instance, and you want to be a volunteer, you can participate in the training. So there are two types of training: basic stranding response training and the more advanced, the medical management training. It's actually a 2 to 3 day training. So lahat ng aspects ng response ituturo sa inyo. Okay, so we have experts from the academy, experts from the BIFAR, from BA who are helping. Okay, So, pag natanggap mo yung kit, anong laman nun? Huwag matakot. After the training, of course, you have the manual. Okay, so we have developed the manual and you have there everything that you need. Okay, so you have package for blood sampling, for instance. So, if you want to get blood, this is what you're going to do. Swabs, serum, etc., etc. So, due to limited resources, we are also prioritizing the types of tissues that we need. But it is our long-term aim to be able to come up with a biorepository for these cetacean tissues. Okay, so as I've said, this is a sample manual. So we keep on improving the protocols. And also depending on the research trust of our students. Say, there is a new student advisee who wants to study this particular aspect, and that student needs another type of sample. So we're going to add that in our collection protocol. So here are some photos from biological material collection training. So it's an actual necropsy training. Okay? So you're also trained, they're also trained to accomplish the so-called necropsy report. So depending on the condition of the stranded animal, there are certain pool of biological materials that you can collect. Of course, if it is a live strander, you cannot collect the kidney. Okay? So minsan nga may estudyante, kailangan niya kidney sa thesis niya. So, ba't parang malungkot ka na pinakawalan yung dolphin? Kasi po nawalan ako ng kidney na sample. Pero masaya naman po ako. Okay? O pag necropsy, namatay siya, necropsy niya, masaya ka? Hindi po, malungkot rin. Kahit may kidney kasi namatay yung animal. Okay? So, roller coaster ride ba nagtithesis ka with marine mammals. Okay? So, I will be sharing with you research highlights coming from our samples up to December 2018. You can see there the points, okay, where we have collected the tissues, and the different colors also represent different cetacean species. You can see how diverse our samples are, okay, and they are coming from different parts of the Philippines. Okay, so we are using cetaceans as sentinels of emerging marine pathogens. Emerging because these pathogens were previously unknown to affect cetaceans, or if they were known before, they are now affecting new populations in new geographical regions. So for this, we are focusing on leptospira detection. Of course, you're familiar with leptospirosis, okay? So we are also monitoring our cetacean species for leptospira and Toxoplasma, Gondii, okay? These are predominantly terrestrial pathogens, and they are emerging as marine pathogens, okay? For Toxoplasma, for instance, we have the cat as their definitive host. But how is it now that they are prevalent in marine mammals? Okay? So for our leptospira detection, we are using the urine and kidney samples. And we have ongoing bacterial culture assays, histopathological analysis, serological assays, using the ELISA, IgM detection, and of course, molecular detection using gene-specific PCR protocols. Okay, so we have tried to know the phylogenetic relationship of two of our isolates compared to the control for leptospira. So we have two isolates. 4K T1.2 was isolated from a Fraser's dolphin, and the 8KT1, I'm sorry, medyo mali, pero kabisado kasi yung name of isolates. That one was isolated from a melon-headed whale. And they are most phylogenetically related to a Leptospira interrogans. So, it's actually pathogenic. there is actually an updated phylogenetic tree for this. Hindi ko lang nadagdag sa slide. And we have also conducted experiments to determine the survival of the isolates in different concentrations of seawater. So we've used 1%, 3%, 5%, 7%, and 10%. And in general, they are able to survive in 10% seawater for 12 days in the lab. So that means they have adaptations to live in marine water. For Toxoplasma gondii detection, we are using the blood as sample. We are also using cardiac muscles, brain muscles, and skeletal muscles to look for disease. Okay, so that's histopathological analysis. We are also detecting IgG antibodies against Toxoplasma using the latex agglutination test. And of course, we are using molecular detection protocol to confirm either acute or chronic infection. So we are targeting the B1 gene and the R1 gene of the parasite. Okay, so we have some interesting results. For instance, one of our detected Toxoplasma gondii from another Pacers dolphin in Santa Cruz del Sur, okay, was found to be closely related to a Toxoplasma which was isolated from a pig. So that means what we are detecting are terrestrial in origin. Okay, so we need to reflect on how these biological pollutants are entering marine ecosystems. So this is based on the GRAS6 gene phylogenetic analysis. GRAS6 is the dense granular antigen protein. Based on the genotypic analysis, we have there another isolate which we found to be phylogenetically related to a strain isolated from human in France. And another one isolated from chicken in Brazil. Okay? So right now, we are suggesting that one of our isolates is an atypical strain. This is in terms of the clonal lineage of the Toxoplasma. There are three main clonal lineages for Toxoplasma, lineages 1, 2, and 3. Then we have Africa, and then we have atypical lineage. So hindi kami pumasok doon sa apat. So it's an atypical lineage, or it could be a new genotype. That's why we're very, very interested to elucidate more on the type of Toxoplasma gondii. Actually, I have another student who worked on Toxoplasma gondii in pregnant women. Okay, so that's the first time that the protozoan parasite is being detected among pregnant women in the Philippines. Toxoplasma detection is actually included in the so-called TORCH screening for pregnant women. T stands for Toxoplasma. But because of limited resources, hindi pa ma-afford ng government natin yung screening. So, we have preliminary results, but we're also concerned on how the results will be used by the hospital. So, if we have detected, for instance, what are you going to do with the pregnant women? Are you going to give medication? Okay, when will the budget come from? Where? I mean, so there are a lot of considerations. But we are happy that we are, providing this information. Okay? So, hirap na hirap din siya mag-sample, syempre, ng pregnant women. You have research ethics. You need to collaborate with the hospital. So, sabi ko, sige, mamili ka. Cetaceans or pregnant women. Pero wala naman akong choice. Okay? So, sabi ko nga, it's a lifestyle if you want to join my love. Okay? So, we are also using our cetacean species as sentinels of antimicrobial resistance. So, we are isolating bacteria from routine sites. By routine sites, we mean blowhole swabs, anal swabs, genital swabs. Okay, blowhole condensate. Ang hirap kunan, syempre, ganito kalaki sa room, di ba? Or dalawa ng size mo. depende pa sa mood ngayon. Siyempre, baka masika ka nito eh. Okay, you need to consider the disposition of the animal, the condition of the animal. So, pag okay na siya, that's the time that we sample for bacteria isolation. And I think, you know kung gaano kahirap kausap ang mga bacteria, di ba? Ate nila. After enrichment, we need to isolate them, we need to incubate them. So a lot of samples getting wasted, for instance. After 3 days pala, kakarating lang sa lab. So we are also innovating, you know, with coolers. Okay, we want, what do you call that? Small incubators that we can bring in the thing, etc. So for this, we're doing the usual enrichment, isolation, purification, phenotypic, molecular characterization, and of course, antibiotic sensitivity testing. So far, according to my students in the lab and research associates, they have 163 isolates coming from wild stranders. What do we mean by captive stranders? Those stranders that are being rehabilitated. Okay, and another set for wild stranders in 2018. So we have 163 isolates being maintained and studied in the laboratory right now. So you can do a lot of things with those isolates actually. So in general, these are the genera of the bacteria that we have. Most of them E. coli, okay? Then we have significant proportions of Pseudomonas and Enterobacter. And the resistance to antibiotics are being tested. So I have here some detailed information, but in general, we are using many antibiotics. So the decision to use those antibiotics was based on extensive literature review. Okay, so what are those antibiotics used elsewhere, for instance, in the US, in UK, for instance? What are the antibiotics being used in aquaculture systems in the Philippines, in poultry systems in the Philippines? They are using antibiotics for prophylaxis, for growth promotion, and it's actually promoting antimicrobial resistance in the marine environment. So we want to correlate our findings with what's happening in the Philippines. So we are also tracing the sources of resistances. Maybe we can see some patterns, but we are also considering that some of our species are migrating, so it's really difficult to track the source of infection or resistance. So this is another ongoing effort right now. So we are detecting for susceptibility patterns and antibiotic resistance genes for Enterobacteriaceae and non-fermenter gram-negative isolates from our secretions. So nagsimula na rin tong research effort na to. Then also we're doing histological assessment. So we respond if the individual is qualified for necropsy, so patay na siya. So you can, the collaborating vets will conduct gross pathological exam. And we are asking for some samples. We collect the samples, we section them, fix them, and then do the microscopy work. Okay, so some of our results, we have detected Toxoplasma cyst in cardiac muscles, in skeletal muscles. And we hope to find cysts in brain samples. Ang hirap-hirap lang din. Minsan, sumusuko na yung nasa field. Di na po namin kayang kumuha ng brain sample. Kasi gabi na po. 24 hours na po kami hindi natutulog. Okay? We have also detected some sarcocystis. And we actually want some students to study this. Okay? So we need a lot of information regarding the parasites of marine mammals in the Philippines. And marine mammal science in general. Okay, lack information on the prevalence of parasites. Okay, so we have also corroborated some of our findings. It's actually exciting because in the lab, you know, you have a group of students working on antibacterial resistance. group of students working on Leptospira, on Toxoplasma, but then they coordinate and corroborate the findings. For instance, researcher working on PCR detection will say, this sample is positive for T. Gondii. And then, my other student working on histological assessment will say, I have seen cysts. I have seen Toxoplasma cysts from the cardiac muscle of your individual. So that means there is toxoplasmosis. Then all of the students can, coordinate, participate in manuscript writing. Okay? So it's good that they sit down together, participate in workshop, and do the matrix. Okay? Correlating data and findings. We also have this exciting finding. Okay, so we have the individual positive for Leptospira IgM antibody, so it's a recent infection. One of the researchers actually cultured the bacteria from the kidney, and another researcher detected Leptospira-associated lesions in the kidney. So you can see there thickening of the basement membrane in the glomerulus, okay, and you can see here some hemocydin fragments, okay, which may be caused by a bacterial infection. So because of this nationwide system of responding to stranding events, collection of tissues, transport of tissues through the years, we have collected many tissues. And these tissues are actually available for research purposes. You just have to coordinate with us. Okay, so my lab is specifically working on Toxoplasma detection, Leptospira detection, antibiotic resistance, and histopathological assessment. There is another lab who is handling the tissue collection. But all the researchers in my lab are, of course, involved in the tissue collection because you need to coordinate with those in the field. Okay, so I want swab, I want blood. Okay, how do you want us to collect? Aside from the fact that you have given us the manual, so those are the usual questions that we get from the field. Unlike nung ako talaga pumupunta sa field. That's why I tried my best to help in the establishment of this system. Okay? Wherein other set of researchers will go to the field, and then there's a system for them to send us the tissue samples. Kapag walang-wala na, pinapadala sa mga bus terminals. Okay? So, kailan ang dating ng Florida bus? Kailan ang dating ng DLTV bus? So, wala ka sa site, pero puyat ka, nasa bus terminal ka, waiting for the samples. Then, takbo ka agad sa laboratory. That's why you need to, file for permits. Okay? So, may advance permits ka na because you can't predict a stranding event. Hindi mo siya pwedeng i-predict. Okay? Minsan, monthsary nyo. Okay? Ng boyfriend or girlfriend. Ay, hindi pwede mag-monthsary dinner. May stranding event. Pumunta ka doon. Okay? And na-experience ko yan. Wedding anniversary ko. May flowers ako and dinner. Wala, sorry. I went to Bulinaw because there is a stranding event. So, 2 days after, lanta na yung flowers ko. Okay, you know that love for science. Okay, so we have this tissues available. Okay, so we are preserving tissues for researchers. Negative 80 to. You can see there the frequencies. And actually, there are certain tissues fixed. That's not formalin, that's actually buffered formalin because we don't want to destroy the RBCs for histological assessment. So we are using a lot of reagents. So this is a general list. Okay, so we also have DNA extracts. So you can detect a lot of things. Okay? With DNA, you can do many things. Okay? And then, we also collect parasites with tissues. Tingnan niyo naman, ang ganda-ganda ng nematodes and cystodes. Diba? So, sayang naman So, kahit wala akong mga students na nag-work doon, kinokollect na rin namin. Malay mo later, merong maging interested. Sayang. Okay, every stranded individual is a precious sample. Okay? Then, these are Phyllobotrium delphini samples. This is actually urinary bladder. Punong-punong siya. Okay? So, the biology of this parasite is still unknown. Okay? In marine mammals. And you can see here, okay, delphini. Okay? In the bladder. So, I always remind my students that you should be thankful to the collaborating veterinarians. You should treat the samples as special also. You are not to waste any serum sample, any blood sample. So just 2 weeks ago, nag-beast mode ako because the serum sample was not properly stored. Okay? So, if you don't store your tissues properly, so you're not going to arrive at an objective result. So you need to repeat the test. What serum sample are you going to use? You still need to wait for another individual to be stranded. Actually, kapag may stranding event, hindi naman kami masaya. Malungkot kayo. Ay, may na-strand na naman. Okay? Hindi ko lang alam, baka masaya yung mga students ko pag may na-strand. Ako lang yung nadulungkot. Oh, may sample ka na, may sample ka na. Okay? And also, our collaborators are involved in the rehabilitation. So, yun lang, kung gusto ng students mag-participate, pero kasi kami busy sa lab doing the microbiology work. But we also have this rehabilitation efforts. And speaking of rehabilitation, they told me that they are calling for volunteers, okay? So, this is, you know the name of this? Si Carol.
Lalaki na po siya.
Lalaki na siya. Dati kasi Carolina siya. Tapos, nag-text sa akin si Sandy last week. Okay, lalaki pala siya. So, yung swab na pinadala niya sa akin ay panglalaki pala. Okay? Even species identification is very difficult. Sometimes we need to call other experts abroad. We need to, you know, send a lot of photos. Okay? So this is in Pangasinan. So they're looking at a possible gut obstruction due to foreign body ingestion. Okay? And another one being rehabilitated, rhesus dolphin in Bataan. Okay, it's currently in rehabilitation pen in Subic Bay. Tingnan niyo yung tail fluke. There's a serious injury. Okay? So, if you want to volunteer, you can get my number after my talk. They need volunteers there, 24/7. Hawakan niyo yung dog. Hindi siya, sinabi ko kaagad. Hindi siya maka-maintain isa ng buoyancy. So, tender love and care. So, syempre kapag stress siya, alam mo naman isabay mo, pakukunan kita ng blood. Okay. O kunan kita ng swab. Hindi, maghihintay. Hanggang stable na yung condition. We have also some success stories. Okay. So, last year, 3 yung ibinalik sa dagat. One of which is Tala. It's a Stenobradenensis rock dolphin released back to the sea on August 21. So, almost a month after rehabilitation. Umiiyak lahat. Yung nakita niyo ba yon? Hindi tayo nag-volunteer. So, chance niyo na to. Okay? Mag-volunteer. Okay? So, may kwento ako about Tala. Okay? Si Tala is actually positive for IgE Leptospira antibodies, but we're still testing more samples. And we've also detected IgG antibodies, and that is important because Tala has been observed to expel placental tissues Okay, so one of the indications of toxoplasmosis is abortion. But of course, we cannot yet conclude on that. So we need to do more tests. So we have a suspected abortion in the case of Tala. Pero saya niya, bumalik na siya sa dagat. Okay? So kahit isang individual lang, that's very important for marine mammal conservation in the Philippines. At konting-konti lang yung nagpa-participate sa effort. So we need more researchers, we need more volunteers, and all these efforts, of course, you know this, okay, contribute to the advancement of marine mammal science in the country. Okay, so thank you. If you want to get copies of the papers related to the study of cetaceans, you can just email me. And I also want to thank my students in the lab. Okay, so we have PhD and master students in the lab. Edwin is also a lab member. He is a research associate. Edwin, pakita ka naman. Okay. But he's working on what? On humans, not dolphins. Okay. He's into forensic biology. Okay. So we have also BS students doing the histological work. And we are actually doing histopathological assessment in collaboration with the VetMed here. So you know Dr. Masangkay. Okay. Okay, so thank you. Okay, so if there are questions, you can just